what you okay what you produce. So today we have two speakers. The first is Dr. Bert Neighbors, who is a professor in the Department of Neurology. He's a neuro-oncologist. He started his career in the Navy, got training in the U.S. Naval Academy, and I'm actually not sure exactly how the residency works in the naval system, but here, okay. He is the head of the neuro-oncology program within the Cancer Center. He plays an important role in the CCTS that I hope he'll take a moment just to describe. One of the goals of this whole series is to make sure people recognize the resources that are available here on campus. And he's going to describe today basic features of phase 1 and phase 2 clinical trials and how those are designed and function.
Good morning, everybody. So feel free to stop me if there's something confusing or we need to touch base on it. As Dr. Chapman said, I'm trained as a neurologist and I practice neuro-oncology here at UAB. So I've been here 23 years. And one of the things that I have learned is that there are a lot of resources, and it's trying to find the resources and get connected. That takes about 23 years, just to let you know. It is a process. There's a lot of collegiality, a lot of interactions. CCTS has kind of provided a platform where a lot of this opportunity can actually be found. So I'm going to talk a little bit. Dr. Oster and I are going to do kind of a 1-2 for you, and I'm going to talk a little bit more on the clinical trial design, and then I'm going to give you a couple examples on the experimental therapeutics side. Now, unfortunately, it's reflective of what I do, which is more neuro-oncology based, but I think you can take something from this and apply it to the projects that you're interested in, no matter what that area field. We've been involved in projects that extend from clinical therapeutics to nutrition studies, and Dr. Oster and I both were on the committee of a nutrition graduate student who just completed some really nice work in neuro-oncology. So it's broad, but a little more of what I show you is going to be kind of specific and reflective of what I do, and then Dr. Oster will speak to you on the biostatistical part. This is critical for your projects, critical for any grant that you submit, that you either have some expertise yourself in statistics or you have collaborated or you are co-investigating with a biostatistician. So on the clinical trial side, just as Dr. Chaplin said, we're talking about human experiments, to be honest with you. These may extend a wide spectrum from experiments with humans that are therapeutic, meaning we're actually doing something to them, to experiments that may not be therapeutic. We may just be collecting information and making observations. There, just to orient you to these phases of clinical trials, so you're familiar with these terms. Phase 0 can also be called a pilot study. There does seem to be some nuances related to this terminology. A laboratory scientist and I recently put in a human clinical trial to the NCI, and one of the reviewers was really fixated that it wasn't really a phase 0, it was a pilot study. So to me, these are fairly synonymous types of terms. Phase 1 usually is a dose finding with toxicity as the primary objective. Phase 2, efficacy. 3, comparison. That's where we're beginning to try to get data that would support market approval. And then phase 4 are typically post-marketing types of studies. So we don't see a lot of those at places like UAB, but they are important because they're collecting data relative to populations that may not have been included in the clinical trials. So clinical trials are often really restrictive in who they allow entry. And so often you wouldn't have pregnant women in clinical trials. You may not have older people. You may have a fairly homogeneous racial or ethnic population. So you may not know some things that once it gets general approval and gets wider use are going to become obvious. So these are kind of terms. Let's dig down just a little more closely and into these. So phase 0 or pilot, basic goal to me is proof of principle. So this is usually there is a biological observation and you want to prove that this exists in a human. The doses we use are usually pretty limited. Often it's just a single dose. Sometimes there may be more than one dose, and there may be what I would call a high dose and a low dose, so kind of a wide distribution in those increments. But usually this dose that we're going to use comes from preclinical studies. So this could be typically laboratory animal types of studies. These usually are sick patients, particularly in cancer trials, often heavily include pharmacokinetics, so we're understanding distribution of drug in a human. And then the purpose of these is to provide us with pilot data so we can consider and use that information to determine how we're going to go forward. So I actually really enjoy this kind of research, to be honest with you. Really typically involves close collaboration with other groups like laboratory scientists. Work we did in nutrition was more of a pilot type of study. So I find these very nice projects, one that you can take on, you can complete within some period of time. It's not like a phase 3 study that may take years to actually do. And so you can advance your particular interests and work with this type of a design. Now, phase 1 studies, these are often in situations where there's a limited amount of human experience with a drug, or it could be, in my case, there's not a lot of experience in patients with brain cancer. And so even though maybe it's a drug that's got wide experience in some other form of systemic cancer, there's not a lot known in brain, so we don't have enough information about the biodistribution and how that may affect CNS levels. So the goals of these studies are usually to define the maximum tolerated dose. This is a bit of an older term, but it's present and you will see it. It may be a term that has more application to oncology, the philosophy in years past always being, well, let's try to give somebody the most amount of a toxic substance that we can. And that sometimes maybe not be the right thing to do, to be honest with you. And so we're seeing, particularly with advances in therapeutics that are becoming more biologically based, we're looking for the optimal biological dose, the dose of the compound, the agent that has that biological effect in a human. Again, the starting dose usually from animals. We don't escalate the dose in that same patient. So once you get started on a dose, you stay at that dose. But different groups of patients do get different doses. And there are different ways these dose changes can be calculated. Some are very small alterations. Some may be larger, particularly if there's other human experience with the drug. Again, sick population, pharmacokinetics now are extremely important because we're having different dose levels, and so we want to see the behavior of the drug in a human across those different dose levels. You can sometimes see different disease types, and so an example may be in systemic solid cancer, where it's all systemic solid cancer. So you may have lung, colorectal, breast patients all in that study. Efficacy is not an endpoint, meaning does this drug work? It's really trying to determine these doses and describe the safety and toxicity before we would proceed to a phase 2. So what's a phase 2 study? So phase 2 is kind of the beginning of when we're trying to decide if this actually is efficacious. It has some type of effect on the patient that's positive relative to the underlying disease state. We are going to continue to collect toxicity data. Even biological therapies have toxicities, often toxicities that were not detected in the preclinical or the animal studies. So the dose is going to come from the phase 1 study. Usually don't change the dose. A lot of times in cancer trials, you'll hear of something called a 2-stage design, and that basically means we're going to accrue some number of patients and then look to see if X number have what we want to see, tumor shrinkage by 50%. And if that happens, then we're going to go ahead and accrue everybody. Statistically, and Dr. Oster can refine that, we're going to be able to potentially answer this question. Or, we're going to accrue X, we haven't seen one of what we're looking for. There's no way statistically at this rate that we're going to define this as a successful therapy. We're going to stop here. And basically what you're trying to do is minimize the number of patients that you're putting on a study to do that. Here, it typically is a single disease type. So we wouldn't see groupings of patients with different types of diseases or cancers. Some centers will like to do a phase 1-2, meaning they write the protocol as a 1-2 study, and that's really a timing issue just because getting all of this through the regulatory. And I'm going to show you a little bit about that because I'll be honest with you, when I first began as a clinical trialist, I had no idea. There was no class like this, to be honest with you. And I just kind of waded in and stumbled around, and so you didn't recognize the components of all this required. Even now, it's kind of hard to keep up with all the moving parts to this, and they're changing all the time as we try to comply, with rules and regulations. Now, a lot of times I'm one of those sending the email, why the hell do I have to be doing this, to my executive administrator. She ignores them typically, recognizing that's my little venting thing. But the purpose of most of this is to try to ensure that we're, one, not conflicted, meaning there's not some bias that we have, and two, that we're protecting our participants and patients' interests. A lot of these studies in phase 2, there'll be stratification factors, and these are usually in cancer things that are associated with prognosis. Okay, so you may want to stratify people based on age because the reality in cancer studies is younger patients do better. So if you had a study where you randomized everybody that was 25 and then everybody that was 65 wound up on another arm, they're going to do different just because of the nature of their age. So stratification factors are often incorporated. Now, clinical trials, as you're thinking about your projects, ultimately you're trying to test some hypothesis and you need some metric, some endpoint to try to answer that question. And so this becomes a critical element of your trial design. So if we just look at an endpoint definition, this is a result, a condition, or event associated with individual study patients that can be used to assess study treatments. And so some characteristics, this needs to be easy. Because one, there are going to be a lot of patients coming onto the study. So if it's complicated or it's complicated to measure, then there's going to be a lot of variability amongst that measurement. And you may have deviations or error bars that are pretty wide that may minimize your ability to say much about that endpoint. You want it to be free of measurement error. You want it to have internal validity, and by that, it needs to be linked to what you're interested in. So an example could be, and we'll look at one, could be I'm interested in using drugs that block tumor-associated blood vessel formation. This is angiogenesis. So these are called anti-angiogenic drugs. So I need to know that the biological mechanism this drug works through is tied to that biological process of tumor-associated angiogenesis. So an example could be the secretion of a growth factor called vascular endothelial growth factor. So we know if we're inhibiting that factor, it's tied to this process of angiogenesis. That gives us some internal validity. External validity means it can be generalized wider, than that laboratory or preclinical experimental set. And this is really probably where we're more challenged than we realize. And I think this is kind of an exciting area. We all are a little bit different, even within genes for which the encoding is relatively the same. So we all have little variations in our germline DNA. And some of these do influence how we respond to different therapies, how we handle different agents, or our susceptibility to different diseases. And our ability to do that type of genotyping is really becoming fairly robust. Sometimes we need to use a surrogate endpoint instead of something that's a direct endpoint. And example could be, if you're interested in cardiovascular mortality, you could use blood pressure changes. So something that we know, hypertension, that influences CV-related mortality, and you're going to look at reductions in that. Lastly, primary endpoint is the parameter used to determine the sample size of the trial and to make judgments on the overall endpoints of the drug intervention under study. And so this is, as you'll hear from Dr. Oster, becomes the critical determination is how we tie these to our sample size and the power calculations. Now, phase 1 studies, the primary endpoint again needs to be clinically or biologically relevant. Remember we said these are typically safety and toxicity. A lot of that can be descriptive. There's standard criteria that exist. The others are the dose of the drug. We're trying to define the dose that we want to take forward into phase 2 studies. It may be related to toxicities, so we're looking for the maximum tolerated dose, or it may be related to biological activity. There are different types of designs. A very standard one for phase 1 is something called a 3+3 cohort design. Basically means we're going to accrue some group of patients, and then we're going to observe them for safety toxicity, collect their PK, and then we're going to accrue another group of patients. Another one is called the continual reassessment method. I'm not going to go into the details of these. There are times that you may want to use one over the other, and these are the types of things that you can consider, particularly if you're interested in a phase 1 study, in your discussions with biostatistics. And also, I think as you're thinking about specific patient populations and you generate preliminary evidence of data relative to that particular endpoint. Phase 2 endpoints are a bit different, and a lot of what applies across clinical trial investigation or endpoints that obviously need to maintain a clinical or biological relevance. They need to be measurable. They need to occur frequent enough that the biostatisticians can determine power. Now, in cancer trials, as well as a lot of other trials like cardiovascular studies, nephrology types of studies, infectious disease, endpoints can be grouped into kind of 2 categories. One is a response, meaning there is some kind of standard event that you can show there's a change in it. And so in cancer, it may be there's a tumor that's 6 centimeters by 6 centimeters, and a patient gets exposed to a therapy and it shrank to 3 centimeters by 3 centimeters. So the overall tumor size reduced. In HIV, it could be you're following the viral load. And so they've got, some degree of circulating retrovirus in the blood at point A, and intervention occurs, and then you remeasure it at point B. So some kind of a response, something we can measure for which there are standard criteria that have been validated that allow us to say a response has actually occurred. Another, and sometimes finding a response is not easy, so we've got to find some other type of endpoint, and often these fall into the category of time to events, okay? And in cancer, we use these a lot, particularly overall survival. So obviously, in some cancers, that may not be good. If we're very good at treating it, most patients may actually survive their disease. In cancers for which we don't do well, then overall survival can be a very, very powerful endpoint. So like in my cancer, the majority of malignant glioma, median overall survival is unfortunately only 18 months. And so it's a short survival, so often we use this as a primary endpoint in efficacy studies, because if we extend that, then we begin to be able to measure that benefit. Another way, which is a little bit less precise, is called progression-free survival, or time to the event. So patient is diagnosed, they get treatment, and then they're in observation. You're waiting to see how long it is before that cancer comes back. This can sometimes be a little more challenging to measure depending on the disease. Now, in neuro-oncology, remember we talked about you need to have criteria that define these events. So this will exist for many types of endpoints that you look at, particularly related to responses. In this case, we're largely using 3 components to define our response. So these could fall into the category of complete response, partial response, or the disease progressed or it just stayed stable. Now, sometimes stable disease is a victory for us. The components of this are, 1, measurements of tumor size, how a patient is doing neurologically, and then because our disease, when it's progressing, requires patients to take large doses of steroids, we can use that as yet another component to kind of guide us. So if we had seen a complete response, then the majority of the tumor visualized on some imaging study has been decreased. Neurologically, they're getting better. That kind of makes sense. If your tumor burden is getting smaller, then you ought to be getting better clinically, and you would not be requiring steroids. And that contrasts to progressive disease, where the tumor is increasing in size, they're getting worse neurologically, and their steroid dose isn't going down, which would be our preference, but it's either stable or going up. Here's an example of a study we were involved in, and so this is a study with a drug that blocked this angiogenesis growth factor called VEGF. And so there's a number of patients. You can see, like on the top row, patient 1, 5, 9, 13. And although it is hard to see the tumor volume, these are MRIs. They're axial, meaning there's slices coming through the brain in this direction. And so the tumor is defined. It's encircled by this red mark. So these are volumetric analyses. The neuroradiologists have determined the total volume of the tumor before treatment and then after, before, after, before, after. And you can see in general for this particular drug under study, there was a pretty robust response, meaning the tumor volume had a marked decrease during the course of treatment. So an example of a response. Now, we talked about these time to events. So survival is a very common one in a lot of clinical trial situations. So it's easy to measure. It's a binary outcome. Unfortunately, you're either alive or you died. It's easy for us to measure it, meaning we can get a date of death and we can get a date that you were diagnosed, and we can look at that time interval between. It is believed, particularly by the FDA, that if you do something to a patient that improves their survival, then that is powerful. That means that particular therapy altered the biology of that disease. It can be confounded, though. So in a number of diseases, when I think about infectious diseases, oncology, often patients may have multiple therapies. So you get an initial therapy, the disease comes back. You get another therapy, the disease comes back. You go on a clinical trial. Ultimately, your survival is the culmination of the effect of all of those different therapies. And then there are what we call variants, just terms for you to be aware of that we'll talk a little bit about here, particularly with progression-free survival. And so this is often defined as the proportion of patients alive and progression-free at some period of time. In this case, 6 months. Could be 3 months, could be 12 months, could be 24. For brain cancer, we've got a historical database, so we know roughly, what that ought to be. So we can often do a single-arm phase 2 study, and we can compare those results to the historical database. Unfortunately, it's 15% of patients are alive and progression-free at 6 months, or the progression-free survival is unfortunately 9 weeks. So when your cancer comes back, it's usually 9 weeks before it grows again, no matter what we do. And so this can serve as a metric. The downsides to this are it is historical. So just as we get better in all aspects of medical care, people do better even if the underlying treatment doesn't really change relative to the disease. We just get better at taking care of people. And so they may do better irrespective of the underlying treatment. So if we just followed brain cancer survival over the past 2 decades, it's improved despite the treatment being exactly the same as far as the foundation. suggesting that we're, one, better at diagnosing the disease, and then just better with the day-to-day care of a patient and dealing with complications related to the disease or the treatment of the disease. Consideration of biological endpoints is important. In this, I particularly kind of focused on maybe anti-angiogenic agents, where you're going to consider tissue effects, maybe effects on the blood, such as circulating endothelial cells, or imaging types of effects. Dr. Oster may comment on this. The incorporation of biological endpoints, they're hard to use as a primary endpoint because unless you have a lot of preclinical laboratory data that allows the statistician to get a good foundation of what we can expect or what we would think we would expect, they can typically require much larger sample sizes than our more traditional endpoints related to efficacy, like time to an event or a response. So another aspect of clinical trials to just kind of touch base on and educate everyone, clinical trials are monitored, and they're monitored by a number of different entities. This is critical in the execution of the clinical trial. If you're involved in a clinical trial that's sponsored by, say, some pharmaceutical entity, it is critical that you, as the PI, meet with that sponsor routinely. They will come to do visits, to review our documentation, and during those visits, a lot of PIs may feel like they don't need to meet with this particular person. That's the representative of the sponsor. They're the one who are going to tell you what you're doing good, what you're doing bad, what you need to improve on. I always meet with the monitors. I would describe, even if we don't really have anything to talk about, if it's just a matter of shaking hands and them relaying everything is well, no issues. Monitors look at toxicities, meaning what's happening to our patients, deaths among study patients. Deaths can occur that are not related to the underlying disease. People can have other events that can happen. How we are doing as far as accrual, often clinical trials have stopping rules, meaning if something happens or X number of these certain events occur. For cancer, the National Cancer Institute has some common toxicity criteria, and so these, an example may be how we grade a rash. So a person is on a clinical trial, they're taking experimental drug X, they get a rash. There are different levels that we grade that rash, and these criteria are defined by the National Cancer Institute. Like I said, different levels of monitoring. Data Safety and Monitoring Committee may be required. Here at UAB for the Cancer Center, we have an internal one. Obviously, the institutional review boards, which are mainly concerned with patient safety, are critical. And then whoever is the sponsor also monitors. So this could be National Cancer Institute, it could be the NIH, or it could be the pharmaceutical company, or a lot of pharmaceutical companies will contract with a CRO, which is just a group that will perform the clinical trial for them, and so they may be the monitor. Here is just a little algorithm where I tried to, put through the steps that can go into a clinical trial development. There may actually be more things to consider. Often we're needing to generate a letter of intent. Usually this occurs after we've had some interactions, at least at my end, with a laboratory scientist, maybe somebody in cancer cell biology, neurobiology, who's done laboratory works, got some interesting data. We want to try to move that forward into a human clinical trial. So we may send a letter of intent. In our case, often these would go to an entity at the NIH called CTEP, Cancer Therapy Evaluation Program. If it's approved, or they say, well, looks interesting, but we need to see this, this, and this, we will ultimately try to get that data or information for them. Get the letter of intent approved, we would begin to generate a protocol document and a consent form. Often you're trying to think, well, how are we going to pay for this clinical trial? Some clinical trials may not require a lot of funding, meaning it may be some variation of a standard of care type activity. So you don't need a lot of funding, but obviously there's specific grant mechanisms through foundations, through the NIH, that can help us with that. Here at UAB, if it's a cancer trial, as well as most within the Department of Medicine, you would submit this to a disease committee. In our case, it's an entity called the Brain Tumor Working Group. We review that, we grade it for scientific priority, and then it goes for a more institutional scientific review and then submission to the IRB. Along the way, you may need to generate an IND, an investigational new drug, if you're using a new drug. If this is a drug that's approved by the FDA for something else, then you can ask for an exemption. That's pretty easy to do. The CCTS actually helps you get an IND if you need one. So Penny Jester and the CRISP group have the resources to help you do that. Once you get the protocol open, it has to be registered with clinicaltrials.gov. This is a big requirement. The institution can be financially punished if you don't do that, and you can be professionally punished if you don't do that. And again, the CCTS can help you with this. Their website is a challenge to manage. So having folks like Penny and her group able to help us with that makes a big difference. All right. So that's a bit of an overview. I'm going to go through a couple of examples of early phase clinical trials we were involved in as kind of examples. Again, I'm a neuro-oncologist, so these are related to brain cancer. So be creative in how you think how this may apply, to what you're interested in. So the first one I want to tell you about is a project that we developed with one of the neuropathologists here, Candace Gladson, whose lab had done a lot of research through the years looking at cell surface molecules called integrins. And so this particular clinical trial was using a drug called Silencio, which is an integrin inhibitor. We did this through the NCI. At that time, the entity we were tied to was called the New Approaches to Brain Tumor Therapy, so NABIT. This is its NABIT number. This is the name of the trial. The drug back then just had these letters and numbers is what it was called. It did not have a name of Silencio. But this drug we knew from Candace's, Dr. Gladson's laboratory work, we knew that one, it could block the ability of glioma cells to migrate, and that it blocked the ability of endothelial cells to respond to these angiogenic growth signals from a tumor. So it blocked angiogenesis. And so we also in our study wanted to evaluate perfusion MR as a way to maybe help us determine the right dose of this drug. So this was a phase 1 study. Primary endpoint was look at maximal tolerated dose, but we kind of knew going in it probably wasn't a very toxic substance because it's actually just 5 amino acids in a circle, so it's a cyclic peptide. And then we wanted to assess these biological correlates, of which perfusion MRI was one. It was a phase 1 study. The drug was given twice a week. We started at this dose. We escalated. I'll show you that escalation. We put 6 patients in each cohort and started off with 5 cohorts, but that got expanded. At the end of the day, the age of our patients was, the mean was 50. Their functional status was about 80. Most had had a couple of tumor regrowths, so they had seen chemotherapy. And then when we look at that study population, there's about 51 patients on this study, but the majority, had the most malignant form of primary brain cancer, glioblastoma. So if we just look at our toxicity data, so we were trying to define the maximum tolerated dose. So we started at this dose, we put 6 patients, and then we collected information related to toxicity, and then we would escalate. And so you can see really there was very little in the form of toxicity from 120 milligrams a meter squared to 2,400. So 2,400 was as concentrated as the company could make it. And so we couldn't go any higher than that. But if we look at just the toxicities described that define a dose-limiting toxicity, we can see very little through the course of all these dose escalations. If we We collected pharmacokinetics. We wanted to better understand how the drug was behaving in humans. We knew from preclinical studies the dose that we felt like we needed to get to. That was defined as the Cmax, the maximum concentration of the drug. If we got that amount in your blood, then we would be inhibiting our target based on our preclinical studies. So we were able to show that there was a dose proportional increase in these PK parameters, area under the curve, and Cmax. Seizure medicines didn't affect it, but the target Cmax we were hitting with the lowest dose of the drug. People responded to the drug, so we had some complete responses, we had some partial responses, and then we had a number of patients with stable disease that was stable for really a long time, greater than 6 months. So remember I showed you normal progression-free survival in this disease is 9 weeks. So these were people that were having a response. And when you kind of look at this, you can see that it's kind of all over the place. So some people responded at a low dose, some people responded at a higher dose. The biological effect we weren't necessarily able to sort out based on just where these responses were occurring. Here's an example of a patient. So she's got this left frontal malignant tumor. We can see it here on the axial. This is the coronal section, so a very large tumor causing a lot of mass effect. She goes on to the study and kind of toward the lower end of the scale, was on study for a year, had almost complete response, ended up staying on 2 years, did have a complete response to the drug. You can see this was back in 2003. She's still alive with a recurrent glioblastoma, which that's unusual. That's 14 years from when she was treated. Now, we were developing this parameter of perfusion MRI, and we were characterizing our parameters for this. So largely, we were looking at 2 values we can extract from an MRI. One is we'll call this CBV, which is basically tumor blood volume or tumor blood flow. And so you measure it in the tumor, and then you compare it to a similar area on the contralateral side of the brain. And so if there's no difference, then this ratio ought to be 1. If it's elevated, it ought to be above 1. If it's reduced, it ought to be less than 1. And so we can see in patients who had progressive disease, if we look at these ratios at baseline versus after treatment, they went up. So that makes sense. People who had stable disease had little change in these values. And people who had responses had a normalization of those values. So we believe these parameters to be valid for the biological effect that we were interested in. We did a couple of different things. We tried to use a model to develop this. We also worked with the biostatisticians. This was Gretchen Cloud, who preceded Dr. Oster here, where we looked at changes in these perfusion parameters 16 weeks to baseline, and we looked, was there a statistical association with either the dose of the drug, the maximum concentration of the drug, or the area under the curve. And we did see that for tumor blood flow, there was a significant association with a higher dose of the drug after 16 weeks of therapy for AUC. So that actually helped us, draw some conclusions in this phase 1 study that this drug, silengitide, at this schedule, wasn't very toxic. The pharmacokinetics were linear. We saw responses. We could do perfusion MRI. It correlated with clinical outcomes, and we felt the biologically active doses were above this 2,000 milligram. And so even though we had kind of this traditional phase 1 study, we had biological correlates that we had incorporated into that as secondary endpoints, which helped us better define how to go forward in a phase 2 study of this particular drug. Okay, that's one thing. I just want to check the time here. So about a quarter till, so we'll kind of go through this pretty quickly. So this is actually a project I did with Harry Sontheimer. Harry was a neurobiologist here at UAB, and Stephanie Robert, who was an MD PhD graduate student. And so they were interested in this process. of brain tumor-associated seizures and excitotoxicity. They'd been particularly interested in this neurotransmitter glutamate, which normally the function of an astrocyte or a glial cell is to suck that up. Glutamate is really how neurons communicate with each other. And so if you leave it out there in what's called the synaptic cleft for too long, then it may cause what we call excitotoxicity. And the astrocytes are supposed to suck it up. So keep it well controlled. But what Harry was able to show is glioma cells didn't suck it up, they spit it out. They made a lot of glutamate, put it out into the space surrounding the normal brain. And so his hypothesis was that glutamate-mediated tumor growth, that the gliomas released this to kill neurons surrounding the tumor so that it could grow, and it would cause excitability, meaning seizures, and excitotoxicity, meaning neuronal death. And they actually understood the mechanism by which that happened. They identified a cell surface transporter called System XC that took in cysteine, converted it to cysteine, and ultimately glutathione in the mitochondria, which had a number of biological effects. But to do that, the cell had to extrude glutamate. And so they looked at System XC expression in patients, and they did find that about half of patients with malignant brain cancer, glioblastoma, had high expression of this transporter in their tumor. These are immunohistochemical stains, from the tumor, and this brownness, this brown reaction product is staining for System XC, and they were able to score that and demonstrate that a significant percentage of patient samples, just over 50%, actually had higher levels than would be expected. Interestingly, we can measure glutamate by MR spectroscopy, so we can generate a biochemical peak. One of these peaks, we call it GLX, because the scanner here cannot separate glutamate and glycine, so it's the two together. And so we formulated a hypothesis. They had identified an inhibitor of System XC. It was actually sulfasalazine, a drug approved for rheumatoid arthritis and inflammatory bowel disease. And so we came up with an experiment where we would do MR spectroscopy on brain tumor patients. We would give them sulfasalazine, and then we would repeat the MR spectroscopy looking for changes in tumor glutamate levels. And this is summarized in this particular protocol, which was investigator-initiated study. It was a pilot study to look at the effect of sulfasalazine on glutamate levels as detected by MR spectroscopy. So if we just look at something called the TCGA data set, we can see that if you've got higher System XC expression, and this is the gene for System XC, there's a shorter survival. We were able to show that by MR spectroscopy, and it's this portion of the graph, so that post giving a patient sulfasalazine, if we use this normalized measure of glutamate, just normalizing it to the creatine, we can see that of the 9 patients we treated, some patients had dramatic reductions in this value. We were able to show that statistically that correlated with the patients who had higher expression of the transporter in the brain. So allowed us to say that tumor glutamate levels could be measured by MR spectroscopy, and that these reductions were greatest in the patients who had high expressions of this transporter called System XC. And so at the end of the day, we characterized this population of patients with this disease called glioblastoma as those that fall into the excitotoxic tumors, meaning they're System XC responsive. These grow faster. They have excitotoxic neuronal loss. They have seizures, and they behave in a more malignant fashion, versus those that are not, which are non-excitotoxic tumors and actually confer a bit better prognosis. So this is my last slide. Happy to answer any questions, but there are different phases to clinical trials. Need to be alert to that. As you are being creative and thoughtful in your endpoints, need to discern where you fit into that spectrum. I favor early, meaning pilot and phase 1, and then very important to consider biologically relevant endpoints, invest time and energy into understanding how clinical trials are actually run and managed. That not only is your responsibility to patients, but it helps you be a better clinical trialist and helps you generate data that you as well as your colleagues can believe. And I think that's it. So happy to answer any questions.
So I just remind people that we have our off-site people who need to hear microphone if you have questions.
I just had a question about one of the trials that you were talking about, the one that said you had multiple cohorts. Each cohort still. I don't know if I misunderstood.
Yeah, no, but that you're making a good point. So each cohort got the same dose. So within that cohort, we didn't escalate the dose.
Oh, okay.
And so that's kind of standard, unusual when you escalate within the cohort,
Yeah.
but can be done. Can be done.
Okay.
Anybody else?
Thank you for your presentation. I would like to know, where would you place feasibility studies?
So I put those in pilot, to be honest with you, and this is a pretty broad space, but it's really the better one, I feel, for most of us in academics to be working in, from graduate student, through even faculty. Those are the ones that, to me, are the better opportunity to prove the interest that we have, to test our own hypotheses. They are more manageable, from a patient number standpoint, and then typically more financially feasible for us. Versus, the more higher efficacy type study, which becomes really increasingly an NIH to a pharmaceutical sponsors study. So feasibility to me fits into the pilot category. So we've done a number of studies that I would describe as feasibility studies where we're trying to generate largely preclinical or preliminary data to define some observation that we would then make a decision on that data if we truly want to go forward, in a larger patient type study.
Talking about designing trials, are you really designing like all the phases, or having to put out some description of all the phases, or do you start with phase 0 and 1 and then?
So I rarely, kind of on a personal, operational, I rarely get out of phase 1 it's pilot, phase 0, phase 1. Now I think you've got to be thinking about your endpoints, like the Silencio, we did the phase 1 and we did a randomized phase 2 of just 2 different doses. But then the company, which was German Merck, did the phase 3 studies. And so we participated in that design discussion, but we had much less of a role there. So, I think you want to be thinking about what you're doing at a clinical investigation level, how that, is going to advance your particular interest. But I don't think for the majority of us, we probably rarely think beyond phase 2 type studies.
I just want to remind the off-site people, if you have questions, just unmute yourself and speak out at this point, or type in a question in the chat box.
And the other thing, I think, I just loaded the slides this morning, so we'll leave them on the computer. They can disseminate them to the group, so you're free to have these to review.
So Dr. Neighbors mentioned something called CRISP, stands for CRSP, which I think stands for the Clinical Research Support
Program. Yeah, we got too many acronyms. It's hard to keep up with all.
Penny Chester, who's a nurse in the Department of Pediatrics and really an expert in clinical trials design and operations, is the director of the CRISP. So they offer a lot of services to help people clinical trial. Access the CRISP through the CCTS website. Just look at the clinical files part of the website. Bert, do you have a sense if a new person who's never done a trial before wants to do one, and they just work with CRISP and have it?
Yeah. I think that's
with an established trialist for the first time.
Well, both of those I think are reasonable. Now, it could be that your area of interest there just may not be someone, here at UAB, or the more mentorish person doesn't mentor very well, so you're on your own anyway. But I think with CRISP and the resources available, you could really accomplish a lot. Now, another plug I would stick in for the CCTS is we do have, I can't remember what this is called, but the protocol review, protocols done quickly. What's that little entity we call PDQ? So I've had junior faculty and fellows use that opportunity. So you do get usually a group, and it's probably the more useful, to be honest, because often you may get 2 or 3, I'd say mid to senior faculty who can sit down, hear it with you, and then give you some feedback, then help you go forward with that. But I do think it's feasible if you've got a pretty clear idea of what you want to do with the CCTS resources, particularly Penny's group, which can help you with the regulatory part, if an IND is required, registering on clinicaltrials.gov, and even can provide you with nursing support for doing the study if that's required, meaning there's some samples collected, interviews need to be done or whatnot. And then I'm the medical director of a CCTS entity called the Clinical Research Unit on the 15th floor of Jefferson Tower. So there are nurses and space up there that a lot of people will use, even to a degree, sometimes if it's just interviews, with patients, or if you need some form of a sample collected. And we do a ton of nutrition studies up there. So there's a lot of nutrition work done in that space.
So you don't have to hire your own staff to do these kinds of studies as you get started.
Yeah, I highly would recommend that direction because, personnel are unfortunately expensive, and this way you're getting expertise and not having to consider taking on all that additional responsibility.
Right.
All right, I will stop, turn it over to Dr. Host.
Just before we take a 3-4-minute break, so you should have all gotten an email towards the end of last week letting you know about the startup of a course that is done through an online program called Kaizen that will teach you principles of rigorous and transparency. If you plan to do studies that are NIH funded, having had training in that area is really, really valuable. The Kaizen approach is a pretty painless way to get training. It's 4 weeks or 6 weeks. 4 weeks, yeah. So there's just you get a little bit of online material, you read it. On Mondays they release it, you read it. There's a sort of a testing period where you get questions, answer, and for 4 weeks you're certified, and it's all registered in the UAB database, so you don't have to keep track of it. It's really a great way to get this kind of training. And I think it's still time, if you're interested, to do it now. They repeat it. I'll repeat it again in March, I'm sure. So it's better to do it. And they do it about 4 times a year. Just keep on the CCTS website. Just sign up. So it's out of your way. Great, thanks very much. So we'll take about a 4 or 5 minute break, and then Dr. Oster will talk about the last phase of this.
Very exciting.
I don't know about that.
That's the critical piece. All right, we'll get together.
Yeah, let's
Try to focus on getting the manuscripts so we can get that out. Yeah. New Year's almost. All right, see you, Bob.
I answered you.
Yes, you are. Yes. Yes.
It helps if I have
Yeah, until no one else. I can send pictures. So I know what I don't need you. Joy is there too. Yes. Sure, I don't think it's so I don't know if you have.
But I haven't with numbers yet. So we all have to emails. Yeah. My real name is Statistical. Nobody knows like that here. So what department? I'm an assistant professor at the orthodontic. Yeah. What are you doing? You did 6 years ago. 6 years? I did take my residency as a student. And I came for an official fellowship. Then I did 3 years residency. And the day I graduated, the next day I was fired. Oh, that's so I didn't have to. You didn't do any stuff. I don't know. So you are the MD. What is MBS? And I graduated at another university. I did the same month, and I was actually #1 and then I got accepted next to the second year is my place. Yeah. Yeah. I did like 3 months vacation. It's for just the basic science.
No, that's fine. I heard it really well when I put my earpiece in my computer. So
If I just talk like leave that here.
I don't know how far down you can go and it still work well, but
Just leave it up here.
Yeah.
That's what I meant. If I can just leave it.
You want to hold it though, not put it on your sweater.
Yeah.
Okay. I think it's okay as long as you.
Well, can I do that?
Yeah, you can just.
Can I put it like here or? Is that too close? Okay.
So I'm going to just mention that you're co-director of Bird, but if you can just take 2 minutes to describe when it meets and what people ought to do if they want to.
The Biostat clinic? Yeah, we'll start that way. Okay. Oops.
Let's go ahead and get started. So this part of the session is going to be led by Dr. Bob Oster. Dr. Oster is professor in the Division of Preventive Medicine in the Department of Medicine here at USD. He has a long career in biostatistics. He's particularly interested in cancer prevention and control, but has participated in studies across a very broad spectrum. He's also the co-director of the BERD, BERG, which stands for Biostatistics, Epidemiology, and Research Design program within the CCTS. That's an incredibly valuable tool for you guys to consider using. And he's going to describe briefly what the BERD is and how it operates, but it's basically a free drop-in clinic for you to talk about study design, and ideally very early in the development of a project that you're doing, so that you can make sure that you develop your project in a way that is going to support the endpoints and the goals of your project most effectively. Somebody asked through the chat box, which I wasn't standing next to at the end of Dr. Neighbor's talk, whether the CRISP and the other CCTS resources that we describe here are available to anybody within the CCTS partner network. And the answer is yes. Some of them are easier to access if you're on campus, but all of them we try to make available to you. We could have a BERD interaction online if you need to. But Dr. Oster will describe there are elements of a BERD extended network at many of the other institutions in the partner network who can also provide local help for you if you need it. So I'll turn it over to Bob, and thanks very much.
Sure. So I'll go ahead and start along the same line. We do have a drop-in biostatistics clinic here. It's really I guess you could say it's a BERD clinic because it also includes epidemiology and research design. So we do have epidemiologists available as well as biostatisticians to assist folks. And that clinic meets every Wednesday from 11:30 to 1 o'clock, and it's always over here in PCAMS. It's often in this room, but sometimes we'll have it like in the room back there or one of the other rooms, but we're always in this building. And the help that we offer is free of charge. So, and it's really designed for for investigators who are looking for support at the beginning of when they're designing a study or setting up their research study. I would say that our clinic is more for like a first consultation or the first couple of consultations. Once we start working with someone in the long term, then we usually meet at other times outside of the clinic. So one benefit of coming to the clinic, though, initially is that we also offer free lunch to everybody who comes. So. And I think it makes sense given that it is during the lunch hour. David, do you think I covered everything enough about that? Okay.
Yeah, so just to be totally transparent, so if you do end up developing a real relationship with a biostatistician to do your ongoing research projects, there is a point at which you have to transfer to some way of compensating them actually doing, and some biostatisticians are okay to work collaborating author on papers, but mostly they need to have a little bit of encouragement that you submit on. So, but the idea here is that the CCTS funds the sort of startup project going to help statistician or geology.
And I think regarding long-term relationships, things can always be worked out regarding how that support will be paid for. So, I mean, there's a lot of resources here at UAB. I'll just leave it at that. My intent for this talk was really to talk about phase 1 and phase 2 statistical considerations for phase 1 and phase 2 trials. After hearing Dr. Neighbor's talk, I began thinking, well, maybe I should have broadened this talk a little bit more and mentioned pilot studies, or what he referred to as phase 0 studies, as well as just briefly a little bit about phase 3 studies. So I may talk briefly about those even though they're not included in this slide set. We'll start with phase 1 studies though. The phase 0 or pilot studies, I think it's pretty clear that those are descriptive and exploratory, and they're going to involve very small sample sizes, and they're really they'll be the most basic type of trial or study that you can do. Now, phase 1 studies, what we use them for is, Dr. Neighbors, I think, mentioned the MTD, or maximum tolerable dose. We also use them to determine the optimal biologic dose, which I don't have in this slide. He mentioned that. Maximum attainable dose, or recommended phase 2 dose. That last one is important because phase 2 study, well, at least every phase 2 study I've been involved in or reviewed was, at least in part, based on the results of several phase 1 studies. So having a clearly defined endpoint for the phase 1 study it's critical. There are 2 common dose finding methods we use in phase 1 studies, but we're not limited to those 2. There's actually several that I won't even mention in this talk, but many of those are rarely used, and they're really only used in very specific situations for some very specific, say, conditions or types of cancer research. Most phase 1 studies are multi-center, just because there'll be small patient pools at different institutions where the patients will have that disease or condition, and in addition meet what is usually an extensive list of eligibility criteria. So I think it might be difficult to do a phase 1 study only including UAB patients in that study, unless you're studying a very common type of cancer or very common disease. Phase 1 studies are used to generate hypotheses. So what I put down at the bottom of the slide, I put what phase 1 studies are not used for. Just to be, you may know this already, but just to be clear, they're not randomized, they're not blinded, they're not used to test hypotheses. And they're not going to be statistically sophisticated in terms of statistical analysis, because as we'll see in a few minutes, what we can do analysis-wise will be very limited. There's 3 major types of phase 1 studies, and those are what I call rule-based designs, pharmacokinetically guided dose escalation designs, and adaptive model-based designs. The rule-based designs make no prior assumptions about the dose toxicity curve, and I would say these are the most, or have been the most commonly used phase 1 designs, even if you go way back to the 80s and 70s and 60s even. This is what this is what researchers have been using up until recent years for almost all their phase 1 studies. And they're used to primarily for dose escalation studies. And the next 2 or 3 slides, I'll talk about specific phase 1 designs. But the 3+3 design, to understand phase 1 studies, you really have to understand what the 3+3 design is. That's been the most widely used and most common design. It's not always the best one to use, but it's one I think that most physicians and other researchers are quite familiar with. The second type of design, those are more rarely used, as you can see they require assumptions of you need real type or pharmacokinetic measurements before you even design the study. And you would have to assume that the dose-limiting toxicity can be predicted by plasma drug concentration or similar measures. And those are quite strong assumptions, so that's probably why those designs are rarely used. Then the third type are adaptive model-based designs. And these are where, well, adaptive means adaptive in that we'll design a study, but then after, say, looking at 1 or 2 or 3 patients, the design of the study, the entire design may be modified after that. And then after another 2 or 3 patients, it may be modified again. So we may not always know exactly what the design of the study will be once we, until we get very far along in the study. That one uses toxicity data, and from my point, the third one is important, actually. Those type of designs require considerable biostatistical support for constructing them and then updating those toxicity estimates. So what I mean by that is that after one patient goes through the study, the principal investigator would need to contact the biostatistician, and then biostatistician would do, well, I know it's very general, but would put that data through an algorithm, and that would determine the treatment or maybe the dose of the treatment that the second patient would be on. And we'll get into that a little more in a minute. So that's what I mean by adaptive, that it's not set for, say, the treatment or the dose is not necessarily set for all patients right at the beginning of the study. Now with the 3+3 design, though, this one it's by rule-based. I mean, everything is set and you know exactly how things are going to progress throughout the study. Or should progress, that's what I mean. For this design, we always enroll 3 patients in the initial dosing cohort. And then we observe those patients. If there are no DLTs in those, then we escalate the dose and move on to the next higher dosing cohort. However, if there's just a single DLT in the first 3 patients, then we add 3 patients to that cohort. So that means we're looking at 6 patients on that particular dose of the treatment. Then if there's no additional DLTs in the cohort of 6, we escalate the dose. So what I mean by that is no additional DLTs in the first 3 that we saw and none in the second 3 that we added. And then in that case, we can dose escalate. But if we see 2 or more DLTs, either in the first cohort of 3 or in the expanded cohort of 6, then we have to stop dose escalation there. Now, some studies, they plan for a fallback dose, and what I mean by that is a lower dose that 3 patients may be enrolled on of that treatment, but that's kind of, we might plan for that, but that's not really where any of the investigators hope that we end up with that on the study. And those results would really be used just to see, well, do very low doses of this treatment have any effect on the patient's outcome or not. But it would be unlikely that those results could lead to a phase 2 study if we have to go that way with a phase 1 study. And then many of these are planned well. And I don't remember any that I've been involved with ending this way, but if the first 2 patients that we enrolled had DLTs, then the study might stop there and we'd wrap it up and move on to something else. So, but that's that. Yes.
Is that the DLT, or would that be a reason to stop altogether?
That's how this type of study is planned, the 3+3 study. Yeah. Is what? DLTs can include a whole host of things. I would think so, but I'm not sure that I can answer that question as well as a clinician can, but I would think that a DLT would include death, yes. And so then we continue to proceed with the design, and assuming that the study's not stopped along the way, we'll eventually be able to come up with an MTD in most cases. And that's defined as the dose immediately below the dose where 2 or more DLTs occurred. So, and we would consider that as a, I guess we can say it this way, as a safe dose of the treatment. I mean, yeah, of the drug or treatment for the patient. Now, it's possible that we may plan to look at 4 or 5 doses of the treatment and we may keep escalating and not find a true MTD. And for whatever reason, we may not have planned to study a high enough dose of treatment, but in that case, we would just declare the highest dose as the MTD and move on. We really can't modify the study then and just keep adding higher doses of the treatment. That would have to be a brand new study. Also in phase 1 studies, we tend to, once we have an MTD, we tend to plan for an additional cohort of patients. I've seen many where they've added 6, but sometimes they'll add, say, 12 more patients at that dose to study drug safety and just to get an initial indication of efficacy, just very initial. The next point I've already said, and the second to last point, if you're ever reviewing a phase 1 study and you wonder why there's a certain number of patients given or a range of patients, well, it's very simple to calculate the maximum number of patients that you'll need. It's just 6 times the number of dose cohorts, and that's because we can have up to 6 per cohort, and then plus any additional patients that we plan to enroll for further safety testing. So that's the maximum number of patients we would need for a phase 1 study. And it may end up being anywhere, say, it could be 12, and the maximum may be even 30. I'm just throwing those estimates out, but the accelerated titration design is less commonly used, but I've seen it used in pharma before. I don't ever remember one being used here at UAB though. That's just a modification of the 3+3 design that we discussed. The difference is that the investigators plan for large dose increments, so they'll start with a smaller dose, but then they'll plan with a certain percentage of dose increment as the next dose. The idea is to accelerate more quickly up to a higher dose than just the traditional 3+3 would allow one to do. And I think what I'm trying to say is this design, the 3+3, they're intertwined designs. This one just, as I said, allows us to test higher doses more quickly. And then an even less commonly used design is a pharmacokinetically guided dose escalation design. And the reason is that given in the first sentence there, the starting dose is determined by animal data that were collected as a standard. And at least in my experience, that's rarely done. Of course, animal studies are, I think, always done or almost always done before drugs are taken into humans, but the starting dose in this case would actually have to somehow be based on results that we obtained from our animal studies. Sure.
So I'm kind of surprised with the 3+3 design. I'm animals, and we have a power analysis where we values, which is usually the p-value of 0.05, take the effect size and standard deviation, and then we calculate standard deviation usually from the data set that we have collected before, and that's already published. Available. Then we calculate the n number of times for a preclinical study, and this 3 just seems like it's a, and I assume this is for these are humans and not the design. But was there any reason why you're not was it?
I will say that there wasn't a statistical reason for it. I think it was based more on experience of investigators over years and maybe decades of research that design happened to work better than others of that type. I will say, though, that the 3+3 design, that modifications of that are often made in studies. You mentioned 5. I think I did see 1 or 2 where actually they used a number different than 3. And as a statistician, I can't say that that's incorrect to do so. I mean, that wouldn't have a basis for that. We can always calculate the different probabilities of dose escalation, though. And so that's kind of the justification I would expect to see in a protocol, especially if it's different from the 3+3 design. So does that help?
Yeah,
Okay.
I'm just interested.
Yeah, in this design we don't. Well, I think the next slide is an adaptive design, but in this design it's fixed, and that's just how we do it. So even if we look at results, we don't change the design later on. So on this side is, I want each of you to at least heard this term, continuous reassessment method, or CRM, because I say it's increasingly popular but not as widely used. This is an adaptive design, as I kind of alluded to earlier, but there's also there's advantages but also several disadvantages of this design, which I think may preclude it from becoming, say, the most popular type of design for phase 1 studies. One I mentioned, which is not clinical, but it requires a lot of biostatistical support. Another thing that happens, again, could be viewed as both positive and negative. In an adaptive design, you would observe a single patient, and you may decide to escalate the dose. Well, not you, but I mean the way the study's set up. You may say, okay, that patient did well. Well, we're going to escalate right now, and we're not going to observe any more patients. I don't really know of any researcher who's comfortable with an N of 1. And but using an adaptive design, that could happen. It could be designed, and it would say, well, after seeing 1 patient, we'll make some decision on dose escalation or not. Some of you may have heard the term of Bayesian analysis or Bayesian principles. All of these adaptive designs are based on Bayesian statistics, which is, they're statistics, but it's almost like it's a different area of statistics that wasn't feasible to use until we had advanced computing power. So to design these studies and to analyze these types of studies is very computer intensive. These days, it's not as much of an issue. I think the issue more these days is that even though these designs exist, some of the statistical theory to back them up hasn't all been developed and fleshed out yet. So, and then the last point. I'm not going to read everything on my slides. I think I purposely created some of the slides to be dense so that you would have the information. But one disadvantage of this design is that it may overestimate the dose for the MTD, and that's not really a good thing either. The big advantage, the reason why people are very interested in this type of design, though, is that it will almost always require less patients to be enrolled than the traditional 3+3 design. So that's the trade-off. So how do we deal with this? This is still, I guess, a work in progress in the field, both among statisticians and among clinicians. But we make modifications to that adaptive design, and we modify that by including some aspects of rule-based design, like maybe taking some pieces of the 3+3 design and incorporating that with a degree of adaptiveness from the adaptive designs. And that would then, that way then we could say, well, we're just not going to have any cohort with only 1 patient in. So we may want all of our cohorts to have at least 3 patients, but with an adaptive design, different cohorts may end up with different numbers of patients, but we could set things up so that each one would have a minimum of 3 patients in them. And I think most investigators are more comfortable with that type of design. And there would be some other rules in there as well, like when the dose may be escalated and when it would not be escalated. And there would be some set stopping rules in there. They wouldn't be completely random or completely adaptive, even though there would be some degree of adaptiveness to them. So even though I might not have said that much about those designs, I think you can get the impression that using that type of design takes quite a bit of time to plan it out and quite a bit of discussion between the physician and the biostatistician. So if you ever want to use that type of design, make sure you see a biostatistician well in advance of the time you want to do a study. By well in advance, I mean maybe, at least 2 or 3 months, or even more than that. Now, let's talk a little bit about sample size justification and then analysis for phase 1 studies, and then we'll move on to phase 2. I have fewer slides for phase 2 than phase 1 studies. I think, as I mentioned, though, the specific design that is used for phase 1 studies is the primary justification for the sample size. So, as I said before, for the 3+3 design, we can compute how many patients we would need based on the numbers of cohorts, for example, and how many that patients will add, that additional patients that we want to add for safety testing. Well, that would be our sample size justification. There wouldn't be a formal power calculation when we're using that design. And I've given an example here that if we had 4 potential dose cohorts and then we wanted to add on an expanded cohort of 12 patients, the maximum sample size would be 36 patients. Now, the clinical investigators, they may know in advance, probably know something about the treatment already, and so they might say, well, we really expect there to be 24 to 30 patients involved in this study, that we don't expect it to stop real early, but we don't really expect to get to the highest dose. So we may just put in 24 to 30 are expected, but we would keep in mind that we really may need 36 patients for that study. Now, for some phase 1 studies, and hopefully I'm not contradicting what I just said, for some of them, though, we can use what I call the confidence interval approach to justify a phase 1 study. And this is not on the slide, but we can also use this approach to justify a phase 0 or a pilot study. So if we're looking on a phase 1 study, we're probably interested in the toxicity rate, but in a pilot study, we may be interested in something else, but what we're often interested in is some type of rate, as opposed maybe to looking for a magnitude of a change in a measurement. And so these are, I think any biostatistician could easily write up a couple of sentences and provide a justification based on this approach. I've given you an example here. The reason I put a specific example in, say, 12 patients is not an unusually small number for a phase 1 study. I mean, there's a lot of phase 1 studies, may I say 12 or 15 patients. So the points I'm going to make is, well, when we have small patient numbers, we would want to justify them using exact confidence limits. And then second, you can see right there, you can see how wide the confidence interval will be. There's nothing we can do about that, but it just gives reviewers an idea of the precision of the estimates, say, for the toxicity rate that we can obtain from our small study. And I know they're very wide, but that's just how these studies are. I put a note at the very end. You can see the width of that first confidence interval is a little over 50%, and the second's around 44%. And yes, that's wide, but that would still provide us with some useful information. I mean, if you're just doing some wild guess, it would be from 0 to 100%, right? That's so at least here we can refine it a little bit and get some idea of the estimates. The precision of the toxicity rates that we can determine from our study. In terms of data analysis methods for phase 1 studies, they're primarily descriptive, so it's always appropriate to, say, compute frequencies and proportions when we're looking at rates for parameters that are measured in serum. We can always compute means and standard deviations as well. It doesn't matter how the data are distributed or how much they jump around. It's always appropriate to compute these quantities. Now, we might question sometimes how meaningful some of them are, but we can always compute them. As I mentioned, we want to use exact confidence intervals if we're computing confidence intervals for rates. If we have adequate numbers of patients in various dosing cohorts, we can actually compare, say, dose cohort 1 to 2. Or say if we have 4 dosing cohorts and we actually end up using that many in the study, we might consider, if it makes sense clinically, we might, say, combine the lower 2 and the upper 2 and do like a low dose versus high dose comparison. Now, of course, there's limits in terms of how we can interpret that, but that often provides useful information in terms of planning for a phase 2 study. I've listed some common statistical tests that you may have heard of there. But when we have small numbers like this, we would probably want to, if we had enough to do a comparison, we'd probably want to use non-parametric methods. And so that's why I've listed the methods that I did there. So that's just something to keep in mind. Okay, let's see. Okay, on to phase 2 studies. Again, these are almost always designed, maybe always designed based on results from that are obtained from various phase 1 studies. We are typically interested in doing some type of comparison for patients that we examine in these studies. Some phase 2 studies do have more than one group. There might be 2 groups. Some may just have one group, and we may be interested in comparing the results of that group to, say, a historical control group of patients that we obtain elsewhere, say here at UAB. When I say obtain, I don't mean we necessarily study them, but we may have data from a similar patient group, and we can use that as our historical control group. Sometimes we may be interested in comparing pre-treatment versus post-treatment status. And sometimes we may... So I think what I'm saying here is that phase 2 studies can take on several different forms, different formats. It just depends on what our research question is. I would say that even though for all studies, including phase 0 studies, we have some type of hypothesis, I think the hypothesis for the phase 2 studies are more clearly defined than they are, say, for phase 1 or even phase 0 studies. And okay, yeah, and I mentioned that even studies that some of these studies may involve genetic testing of participants, and we may have some genetic parameters that we're looking at in these studies too. Sometimes, I won't say always, but sometimes our eventual goal is to move on up to a phase 3 study, and Dr. Neighbors briefly touched on phase 3 studies. But we have to have very convincing results from the phase 2 study in order to move on up to a phase 3 study. Now, in the phase 2 study, we almost always have 1-stage or 2-stage designs. I'll get into that in the next slide, I think. It's possible to have a 3-stage design, but it's rare. And the reason it's rare is because when you have more than 1 stage for this type of design, subsequent stages are conditional on what happens in the first stage. So if we have a 2-stage design, stage 2 is conditional on what happens in phase 1. If we had a 3-stage design, then it would get very confusing because we'd have to set it up to say, well, does this 3rd stage, is that conditioned on what happens in phase 2? Is it conditioned on what happens in both stages 1 and 2? And the very last point, that there really weren't that many ways statistically to handle phase 2 studies until, say, the mid-1980s, when we had a large increase in available computing power. I'm going to mention a couple that go way back, but by that I mean there weren't many methods developed by statisticians before then because there weren't any good ways to, say, come up with and develop and test the methods before, say, the 80s. So when we say a 1-stage design, what do I mean by that? Well, that's the simplest type of phase 2 design. And we just design that study and perform it as planned we don't plan to enroll any additional patients other than what we said we would enroll. We don't plan to create any additional arms, and we don't have any conditional outcomes. We just do the study from start to finish and then analyze our results, and then we're done. The sample size is fixed there, so we fix that at the beginning of the study. Well, I'm oversimplifying it by saying fixed, but by fixed I mean a biostatistician would be involved and we would have statistical justification for that. But then once we have that, that's a sample size we would use and it would not change. So regardless of how the study was progressing, we would still follow it through all the way until the end. The 2-stage design is a very popular type of phase 2 design. One of the reasons is because it potentially it's less or fewer patients are subject to a treatment which may be non-efficacious or even harmful. Whereas if the treatment appears to be very helpful, then more patients will be enrolled into the study. So the purpose of the first stage is to rule out treatments that have little or no biologic activity. And so these studies are designed, and again, the way you see it on the slide here, it's oversimplified, but there's more to it than this statistically. But we would say that there must be a minimal level of activity in the first stage. So, and this varies study by study, but we might want to say, well, if the treatment is effective in at least 20% of the participants in the first stage, then we'll enroll more people in the second stage because we think that treatment is showing some promise, say for some type of cancer, especially for types of cancer that may be less common. I've seen some phase 2 studies where in the first stage, the first stage might be planned, say, for 10 patients, and if the drug shows efficacy in just one, then they'll plan to move on to the second stage and enroll a larger number. For very rare cancers, that may often be the best approach. For things that are a little more common, though, as I said, when I say 20% of participants, well, that might be 3, it might be 4, it might be 5. I mean, it depends on the size of that first stage. And then the second stage is conditional, and of course by that I mean is if we meet the minimum number, or the minimum number who are required to show efficacy in the first stage, then we proceed to the second stage. And the second stage, the number of patients in the second stage may be equal to that in the first stage. It may be, or it could be potentially a lot more. There's been tables developed for this, and then using software we can come up with a very large number of possibilities for these studies. The very last sentence in that paragraph that was important, that the response rate for the phase 2 study, it's based on the total number of participants enrolled in both stages. So when all is said and done, we combine all participants and say that's our patient group, and all of our statistics are based on all of those participants. We don't do analysis separately for each stage. And I've said the other things on that slide. Yes?
I have a question. I'm in the orthodontic department, so we have no drug open. Usually we test some science or treatment system. So I would choose a lot of studies, phase 2 studies either one time or so my question is if there are some trials that have been done before, because there are some out there but not too many patients involved. If we do the same clinical trial that has been published before, do we have to justify why we're again and see what's different, or just go ahead and try to organize our trial, with something I said before?
Can you repeat it so that the online people can hear what the question was about? Summarize.
Sure. I think the gist of the question was that if an investigator is interested in essentially repeating or doing a clinical trial or a research study, it might not be a full trial, that somebody else has done, does justification need to be provided to do that study or trial, or can the investigator just go ahead and do it? Is that?
Yeah, because you talked about sample size, so you have to justify that sample size, or?
Oh, okay. I would say, okay, there's maybe a two-part answer. The first is, I think if it's pretty much the same study or trial, I think some clinical justification is needed for why do you want to do something that's already been done? What will be gained by repeating this? And is it worth putting your resources into doing that study? The second part is, for any study that you do, you need a separate sample size justification. So even if you're using the exact same sample size as another study, the sample size justification should be done separately and should be considered separately. I don't think I've ever seen anyone do exactly the same study that somebody else did. They always add a new twist to it or a new piece to it. Does that help?
Yeah. Because in our field, we're just very limited in what we can do. So, for example, there are people like 30 patients, and they try and see if implants work. And they have some results, and then they go ahead and start using it. So I was just thinking, people will do that again, but maybe some size, and that would.
Okay. Yeah, for those online, the individual asking the question is in orthodontics, and they often study implants and look at, say, dental devices as opposed to studying treatments or some type of internal drug. And I think that's why the question came up. So I just wanted to say that for people online. Okay. So I'll go ahead and proceed then with this. We're often, again, even in phase 2 studies, we're often interested, well, I think we're always interested in the response rate. That's why we're doing the study to begin with. And I would say the response rate is almost always the primary study outcome variable. And I want to come back to something that Dr. Neighbors mentioned because that's not in here, but I think it's important. But first, results are sometimes provided for one or two key secondary outcome variables. And those variables can be of different types. They might be continuous, like say a serum measurement or an external measurement that was taken on a patient with a measuring device. And they might be categorical, which would be like a yes or no type of measurement, or a time to event measurement, and that would be the different survival outcomes like progression-free survival and overall survival. Those are the most two common ones, but I've seen oncologists who are interested in lots of variations on types of survival that they're interested in. But just my experience indicates, though, that those are almost secondary outcomes. Some may be key secondary and some may be less important. But we seldom have a survival outcome as a primary outcome for a phase 2 study. That's based on my experience, though. For a one-stage design, and say that this would probably apply to the orthodontic type of studies as well as treatment studies where there's where future actions or where the later stages of the studies are not conditioned on the first part. For those, we can just use conventional one or two group statistical methods. That depends on how many groups we have. But those would be statistical methods that one would typically learn in their Stat 101 class. So, like the two-group t-test or analysis of variance or non-parametric methods that I mentioned earlier in the slide. All of those would be appropriate or might be appropriate for one-stage phase 2 designs. Now, for two-stage designs, there's a few additional considerations, and some of the. Sorry, I just got ahead of myself. This slide's on sample size justification, so let me backtrack a tiny bit. For the sample size justification for one-stage designs, we would use methods that are based on the traditional one or two group statistical methods. And those might be some of the ones I mentioned. For two-stage designs, this is a method that I'm referred to that I said goes way back. In, say, the 50s and 60s up through the 80s, Geehan designs were the standard justification used. And I referenced that on my last slide. That was a paper published in 1961. And that's just what that was really the primary paper that investigators based their phase 2 designs on for many years until the 1980s, and really until 1989 when Richard Simon's paper came out about optimal two-stage designs. And really since that time, though, most two-stage designs are based on Simon's work and then extensions of his work that have been done by biostatisticians since then. So if you're reviewing protocols, you really shouldn't see Geehan designs used anymore. I mean, I would go further and say that those designs are outdated and probably shouldn't be used in clinical studies anymore. The newer designs just have many more desirable statistical characteristics. And I think they're also more flexible for the clinician, too. Okay, now returning back to data analysis methods for phase 2 studies. Again, my third point there, that's kind of where I was going a few minutes ago, that Conventional methods for group comparisons may be needed depending on how many groups we have in the study. Statistical methods for paired data may be needed if we're looking, say, at pre-treatment versus post-treatment type of data. Now, if we're not doing a pre and post study, then we don't need to consider those methods further. Sometimes, though, you might be a little surprised in that even though that pre versus post may not be our primary interest, a lot of times we'll take baseline measurements on patients and then look at them after treatment and see how they did. And that's kind of paired data, and we may need to handle it as paired data. So it's just something to keep in mind. The methods that we select, which we select before doing the study, of course, those may depend and really do depend partly on the expected final sample size of the study. That's and the number of study arms. Those are the two primary considerations. The conventional methods I referred to, like the ones used to compare groups, well, those can almost always be used. But if we want to do modeling for our studies, like regression models and various other types of models, we need to have a reasonable amount of data, participants in our study to obtain data from in order to even run those models. So we can't develop any type of reliable, or I might even say useful, model based on results from 4 or 5 patients. It's just, I mean, I think we're almost just wasting our time if we try to do that. Now, once we start getting up to, say, even 10 or 12 patients, we can develop some, say, preliminary or some kind of models that we can then at least start the discussion with and see, well, is that something we might want to apply to larger studies? I mean, those kind of models. Okay, I think this is my second to last slide. Let me see, yeah. And I'm doing fine on time. I see I do have a couple minutes left, so I do want to cover this slide. In almost every talk I give like this, or if it's on another statistical topic, I include one slide about statistical software. And that's because I think everyone who does analysis these days uses statistical software. We're long past the days of pencil and paper and using, well, we still use a calculator on occasion for certain things, but for most things we really need to use statistical software to run it with. Now, for sample size and power calculations, there have been a software developed specifically just to do that. Those software won't do anything else except that, but they specialize in this, and they're widely recognized among pharma and among academic folks for their accuracy. So, like when I work with investigators on grant submissions, if there's a little bit of room, like if I can add a few if I'm given a little space to add a few extra words, I'll mention the software that we used to do the sample size computations with. And I'll mention the version that we used too. One of those is it used to be nQuery Advisor, and when we speak orally, we still say that, but technically it's nQuery Advisor plus Interim. And that's because in addition to doing sample size calculations, power calculations, it can also do sample size planning for studies that involve formal statistical interim analysis. So the company that developed that added all of that into one package. The second one is PASS, and that, I think that's an acronym actually. It stands for Power and Sample Size. And they're put out by different companies, and they're not I'm not sure what all of you would consider to be expensive software, but these are not inexpensive software packages. So, I mean, they're not prohibitively expensive, but they might be several $100s, and that would be for an academic license. Of course, for a corporate license, it would be even more than that. As I said, though, they're regarded as being reasonably complete in terms of the variety of calculations that can be performed. So anything that a statistician would want to do with in terms of calculation for an investigator can be done using one of these software packages. If it's not included in these, then there might not be a way to do, that kind of calculation. So that's for that. Now, for statistical software for analysis purposes, the 4 packages I've mentioned up there, SAS, Stata, SPSS, and R, those are what we call the big names in statistical software. There's a number of other software packages out there. I don't know how many, really. There's some that one would pay for, some that are online and are free. But if it's mentioned that, say, we'll be running our analysis using one of these, I think that reviewers will almost universally accept that. They'll say, okay, they're using something that's been very widely used, has been very widely tested. It's known to give accurate results. All 4 of these, well, okay, I've said the next point pretty much. Some software packages, now this is more generally, are easy to learn and use, and then others require a substantial amount of time for one to become proficient. I'll mention one not on my list. I know that the package Prism is popular among clinicians, and I've actually had a couple of them show. I don't have it myself, but I've had a couple of them show it to me, and it really is it may be one of the easiest ones to use. And I think it's at least modern or newer versions, it's pretty accurate. But it's still it does basic things and maybe a few modeling techniques. So if you use that, it's fine, but you'll still likely need to consult with someone like me to run more, say, intermediate level statistical analysis and maybe even advanced level analysis. I would say, though, just again, based on my experience and based on what pharma does, for the last 20 or 30 years, I would say SAS has been the standard statistical software package for statisticians, at least anyway. That's also the one, though, that probably requires the most time to become proficient at, because one needs to learn how to program it. So it's like learning any other programming language. The nice thing about SAS and I think R, those two maybe more so than Stata and SPSS, is that it's easy to reproduce the results. I mean, because one is writing code and saving code, it's easy for somebody else proficient in those to look at the code, to run programs and see, well, are these results appropriate? SPSS is nice in that it's a point-and-click software package, but it's not as easy to reproduce results using that if somebody else wanted to do that. Yes?
I use syntax when I'm using SPSS, and so I've gone back and run the analysis because I saved the syntax. So I find SAS incredibly difficult to use. I even bought the book for dummies, and it's not working. I had no problems with Stata. I haven't tried R yet, and I have worked with M+, which was a waste of because it wasn't much difference in terms of.
Well, that one's a specialized software, though.
Like
That's different.
Used for dichotomous variables, but there wasn't much of a difference
Yeah.
when I looked at results with M+, and when I ran it with SPSS. It just cost me more money. So I dropped that from my toolbox, but SAS,
Oh, that's a fair comment. SAS it's one of those that, yes, that's true, but for those of us who learn it and use it for years, though, we really like it because we have, at least I feel I have control over everything using SAS, whereas with other even, and I know SPSS by the way too, and I've used the syntax before, and I think it's helpful to do that, but even with SPSS in the syntax mode, I don't always feel that I have complete control over everything that the software is doing, but with SAS, I feel like I do, and I think that's because of all of these are good with statistical analysis, but SAS may have the most capabilities in terms of data management. Now, some others may disagree, but just in and that's because you're programming it. SPSS has some of that in syntax mode too. Like if you use syntax, you can type in that. But if you're using someone who's just using point and click, you really don't have a lot of control over. You have control over the analysis, but not a lot of control over the data management and different transformations and looking at subsets of data. And hold on just a second. My last slide contains references, so I kind of wanted to say that was the end of the formal talk, but certainly I'll answer any questions now.
One of the competing packages for R, I hear like there's another Python, like Python and R are like open source, and that's why it's popular.
Which one again? But
Python. Python, Python.
Oh, Python.
Yeah.
Oh, okay.
But I'm not sure how like it's used in academia or not.
That's a good point. I know a few people here who use R, but they also use something else, and they also use one of the other three software packages, which might be their primary analysis package. Yeah, with open source software, I mean, again, the advantages, of course, are kind of what you mentioned. You can program it. People are constantly updating procedures and adding things. And I think usually, I know R is free. I don't know if Python's. Yeah, it's typically free. But the disadvantage is that many of those routines and procedures haven't been widely tested and not widely published on. So you sometimes wonder, well, is it always giving me accurate results in every situation? So I think that's why open source software hasn't been adopted as much yet as the standard software for most people. But I know R. I'm not as familiar with Python. I've heard of it, but I know that R is growing every year, and it seems like in terms of the number of users. So, and also books. A lot of books now have been written on R. So I wouldn't have a problem if somebody else did their analyses in R, say for a peer-reviewed paper. I mean, I wouldn't question it and say, well, no, R is not very good software or anything, but I don't know if that helps or not, but that's just my personal opinion. Other questions on software or anything that we went over?
I wonder if there's any expert here that I can ask questions or learn a little more.
Okay, I'm not aware of any course offered specifically for R. I know at least one and maybe two of are the folks in our bird, though. They've used R to some degree, and I'm sure that they could, if you have questions, I'm sure that they could answer them. I wouldn't be the right one to ask probably, but I could refer you to one of them and they might be able to help you. Any other questions?
Will you be here at noon today?
Yes, I'm here at I'm here at all of the clinics unless I'm either on vacation or I'm traveling to a conference. And I should have said this, but typically there's four of us here that staff the clinics. So like if you were to come and I'm not available, one of the others could also give you expert help. So you don't need to feel that you have to speak to a specific individual. So I mean, unless you want to, but you don't have to feel that you have
I
to.
attended the clinic, so I worked with Dr. Lee.
Oh, okay. Okay, I guess I forgot, but
Changed my hair around, so.
that's okay. Jean, is there any way to find out if the people online have questions or not? Okay.
I just had a question about the stat clinic. There's one on Monday too, correct? I went to one on Monday over at Lister Hill.
Good point. Right.
You can't make.
Yes, and that one's from, I think it's 10 to 2, and there's one. I think there's 2 other Bird faculty there, and there's also some graduate students there that work along with people. So yes, I'm glad you said that. Yeah, that one's on Monday from 10 to 2, and I'm not at that one. I mean, for what it's worth, but my point is there's a different group of us at each clinic so that we can hopefully offer, so that statistical support is offered by the CCTS in different venues, different places. For those on any of those, yeah, any of you who are still online. Oh, okay, that's what I was just going to get to actually. The question you're welcome to leave, by the way, but if you want to stay, the question that's asked online is it possible to call into a CTS bird drop-in clinic? I would say the answer is yes if it's arranged for in advance. We don't have like the things set up where people can just call in at random, but we could take call-ins during the time or even at other times if the clinic times aren't convenient for people. Yeah, and in general, yes, we do. And in fact, I have worked with investigators from some of our partnering institutions. So thanks for the question. Is that Crystal? Looks like CHR. Crystal, okay. Any other questions here or from those online before we wrap up? Okay, if not, then thanks everyone for your attendance.
Thank you. Thank you.
Do I close this? Okay.